Hi guys, one of my learning objectives for you today is to really understand the difference between passive and active transport. Last week we spent a lot of time looking at passive transport, which is a type of cellular transport where particles will move across the cell membrane without the use of energy. Like we said, think of a passive individual, somebody who doesn't have a lot of energy. The two types we primarily focused on were diffusion and osmosis. We also focused on facilitated diffusion. So remember, diffusion was things basically involved molecules and substances moving from an area of high concentration to an area of low concentration. Osmosis was the diffusion of water, and water moves from low solute to high solute. Now there's another type of transport that we'll look at. It's called active transport. This is when materials actually move against the concentration difference or against the concentration gradient. That means they move from a low concentration to a high concentration. That requires energy. It's almost like, think about passive transport. You're riding down an escalator. You're going from a high area to a low area. Because you're riding that escalator, you're not using energy. But active transport, think about if you're starting at the bottom floor and you're trying to run up to the top floor. You have to exert a lot of energy to be able to do that. Two ways that the cell actually does this is through endocytosis and exocytosis. Now if you look at the roots here, endo we know means in, cyte we know means cell. So it's a process of bringing into cell. Exocytosis, exo means exit, cyte means cell, so it's a process of leaving the cell. And I want you to write down this key vocabulary word. This involves vesicles, which are membrane-enclosed sacs that store and transport substances. These are what are used to move large molecules. All right, so as we said, active transport, the movement of substance is against a concentration gradient. So substances flow from a low to a high concentration. The types, as we said, there's endocytosis, and there's two main types of endocytosis. One is called pinocytosis, one is called phagocytosis. Pino refers to liquid, phago refers to food, and then of course exocytosis. If you look at this image here, I'm showing you at the top diffusion and osmosis, where you can see these purple molecules. We have a large concentration of them. They move to a low concentration across that bilipid layer or that plasma membrane there. Facilitated diffusion, remember, it's through the use of a protein channel, that light purple molecule, and you can see the yellow substances again move from high concentration to low concentration with the help of the protein. Active transport, as we said, requires energy, and you can see the yellow structure that says ATP. That stands for adenosine triphosphate, and it's the type of energy that the cell actually uses to move substances from a low to high concentration. So endocytosis. As we said, endocytosis is a process of taking material. This could be fluid, macromolecules, which just means large molecules, and large particles into the cell. This is done by the unfolding of the cell membrane. So if you look down at this picture, you can see the phospholipid bilayer, or the cell membrane, it's kind of making a dip in. As you see in the second part of that image, you can see that that actually closes off and then it pinches off. Okay, it forms an actual vesicle. So the infolding breaks loose, as I said. That vesicle, once it detaches from the cell membrane, we consider a vacuole within the cytoplasm. And if you remember what a vacuole does, a vacuole stores liquid or waste within the cell. There are two types of endocytosis, pino and phagocytosis. Pinocytosis is basically when tiny pockets form along the cell membrane and they fill with liquid. Pino, as I said, refers to liquid, and so the vacuole actually holds liquid substances. And just so you have a correct definition of a vacuole, I'd like you to write down it's a membrane-bound cavity within a cell, and it often contains a watery liquid or secretion, as we learned in cell organelles, maintaining or holding waste or nutrients for later use. Phagocytosis, again, type of endocytosis, This occurs with extensions of the cytoplasm when it surrounds a particle and packages it within a food vacuole. Again, a vacuole which is holding something, and phago means food. I do have just a quick little video if you want to check it out on YouTube. You might want to go there just to see this actually in action. Lastly, here we have exocytosis. This is the process of getting rid of cellular waste, or where the cell might actually secrete hormones. So if you think about the different types of hormones we release, estrogen, males for testosterone, all sorts of different types of hormones are actually released in this way. You release mucus from secretory cells. You'll see sweat released from sweat glands, oil from oil glands. This is all done through the process of exocytosis. And if you look at this image here, it's really just the reverse of endocytosis. So the membrane vacuole surrounds material. That membrane vacuole fuses with the cell membrane. Once it actually fuses with the cell membrane, it releases the substances out into the exterior of the cell. And again, just an image to finish this off. As you can see, endo and exocytosis are really just the reverse of each other.